It is a pleasure for us to welcome Dr. Aimin Xu, who is a professor at the University of Hong Kong. He holds adjoint appointments in the Department of Medicine and the Departments of Pharmacology and Pharmacy. Additionally, he serves as the director of the State Key Laboratory of Pharmaceutical Biotechnology. His research focuses in adipokines that play a crucial role in obesity-related insulin resistance, systemic inflammation, type 2 diabetes, and vascular dysfunctions. Some of his notable findings include identifying the post-transcriptionally modified isoforms of adiponectin, as well as pro-inflammatory adipokines like adipocyte fatty acid binding proteins and lipocalin-2. The title of his today's lecture is Hepatokines in MAFLD from Bench to Clinic. So thank you very much, Professor Xu, for joining us. We are looking forward for your seminar, and I give you the space and the attention. Thank you very much.
Thank you, Rosanna, for your kind introduction. So actually, I joined the University of Hong Kong around 22 years ago. And then I did my PhD in New Zealand. It's in a small country in actually. My lab has been working on these factors released from the liver and also adipose tissue, and then that namely adipokines and the hepatokines, and then to investigate how these adipokines and the hepatokines modulate and then to energy metabolism and also tissue homeostasis, and also how dysregulation of these hepatokines or adipokines contribute to obesity-related metabolic complications, and including MAFLD. So today I'm going to share with you and then the several hepatokines and adipokines we have characterized so far, and then on their role in the MAFLD. So I think this audience, everybody know this non-alcoholic fatty liver disease, and then this is a metabolic associated chronic disease, and then this is closely associated with obesity. And in obese individuals, and there are over 50 to 90% of the prevalence. So this is a major cause of liver-related mortality and the transplantation. And also, so far, there is only one approved drug available for that. So this is so this disease and include a wide histological spectrum ranging from simple fatty liver and then to NASH or MASH that is also characterized by inflammation, hepatic ballooning, and also the steatosis. And then once we get to this NASH or MASH stage, and that is can be further progressed to fibrotic NASH or cirrhosis or even liver cancer. So as I said, because this MAFLD is a continuum of obesity-related medical complications. Therefore, several years ago, and then this was renamed as a metabolic associated fatty liver disease, MAFLD, or more recently, and then this is renamed as a MAFLD. And because this is a close association with a large number of obesity-related complications, especially, so this disease and is this multiple factorial disease and with different etiological origins, including these genetic contributions, epigenetic modifications, environmental factors such as dietary nutritional factors and the behavior factors all contribute to this MAFLD or LAFLD. And many organs, including gastrointestinal system, adipose tissue, even brain and the cardiovascular system, and then they are involved in the development of this disease through such as alterations of gut microbiota, through gut liver axis, and then alterations in adipose tissue dysfunction that is the adipose nerve axis. So they never to communicate with other organs, mainly by secrete the factors, these called the organoids. So this liver secrete hepatokines, and the muscle secrete these myokines, and this bone tissue secrete osteokines extra, and then adipose tissue secrete adipokines. So this regulatory crosstalk between different organs through these organoids play a very important role in regulating tissue, the energy homeostasis, and also the tissue functions. And then this regulation of these hepatokines and adipokines are the key player in obesity-related metabolic diseases, including MAFLD, such as during the obesity. So this lipotoxicity causes responses in the liver tissue and in the adipose tissue, will causes the dysregulated production of the adipokines and also hepatokines, and then act in the different metabolic tissue and contribute to metabolic dysregulation and MAFLD. So my laboratory have been focusing on identify the novel hepatokines and the adipokines involved in the obesity-related MAFLD development. So what we did many years ago, so we have initiated established this clinical cohort from bariatric surgery patients with around 1,200 cases from several centers in mainland China. So we could obtain the different tissues, including liver biopsy, adipose tissue, and the blood. And then on the one hand, so we have done a comprehensive histological evaluation for this, the liver histology, so that spanning from this normal liver, borderline NASH steatosis, and the NASH and the liver fibrosis. And then we did a comprehensive omics-based analysis, try to find the biomarkers associated, mainly the protein biomarkers associated with different stage of this MAFLD. And then we conducted a comprehensive the mechanistic investigation, clinical studies, and also develop assays for potential diagnostic application. So so our lefty samples were independently evaluated by 3 experienced pathologists, including 2 from Canada, 1 from Australia. So, for example, in our case, by using this strategy, so we have identified or characterized many adipokines, hepatokines, so that is involved in this obesity-related disease, including this MAFLD. And we are the one of the earliest groups working on adiponectin in 2003. So we reported for the first time this adiponectin can protect fatty liver disease. And then we also identified this secreted form of adipocyte fatty acid binding protein use our cohort. And then this lipocanin 2, they both play a very important role in linking obesity to metabolic inflammation. So in 2008, so we also discovered this FGF21 secreted from adipose tissue and the liver. So they are associated with obesity-related complications. And today I'm going to focus on how the crosstalk between the FGF21 and the adiponectin contribute to the protection of non-alcoholic fatty liver disease, how dysregulation of this axis and then contribute to MAFLD. So FGF21, maybe many of you are familiar with this hormone. So this is secreted predominantly from the liver tissue, but we also find the adipose tissue can also secrete this FGF21. So this is FGF21 has potent metabolic benefit. So this is the endocrine member of the FGF21, FGF superfamily. So unlike the traditional or classical FGF family, so this FGF21 only has weak binding to FGF21 receptor, and then due to lack of heparin binding property. And then this FGF21 do not have mitogenic activity that they can be released into the bloodstream. So in the last decades, there are many studies in animal models has shown this recombinant FGF21 has multiple metabolic benefits, so including increase the glucose utilization, increase insulin sensitivity, and increase energy expenditure, and also reduce the body weight, improve the lipid profile. So this is a first study published by Aimin in 2009. So they show for the first time, and when the FGF21 recombinant treatment, treatment with recombinant FGF21 can alleviate high-fat diet-induced lefty in mice. So that is, you can see from here, this is FGF21 treated group, and then this already staining show this is markedly reduced in lipid accumulation. So subsequent many studies have found, and on the other hand, if the FGF21 deficiency that can promote liver steatosis, ballooning, and fibrosis, as you can see from here. And then this, so under the high-fat diet condition, so these knockout mice show obvious steatosis and increased ballooning and then skull, and also increase the fibrosis, fibrotic formation. And then there, most importantly, and then there are studies suggesting this deficiency of FGF21 can promote hepatic hepatocellular carcinoma formation to in mice on a long-term high-fat diet, as you can see from here. And then this is the first study published in 2018. So they show in this knockout mice under the high-fat, high sucrose diet, and then after 52 weeks, this developed obvious the HCC. And this is also promote NASH to HCC transition. So through this activation of interleukin-17A signaling pathway. On the other hand, this forced expression of hepatic FGF21, the hepatic FGF21 can delay the initiation of chemically induced HCC. So that certainly can also protect the liver cancer. So we have been working on how does this FGF21 exert its metabolic and hepatoprotective effect, and how to develop these FGF21 based therapies for obesity-related MAFLD and liver fibrosis. And then this FGF21, as I mentioned early on, so these act on target through binding to a receptor complex, including the classical FGF receptor 1, FGFR1. So this is classical one. Because this FGF21 does not have heparin binding domain, so this need a co-receptor called KLB or beta-clotho to form a receptor complex and then initiate the intracellular signaling. While this FGF receptor 1 is ubiquitously expressed in the body, but this KLB1 only expressed in the several tissues, especially in adipose tissue and in the brain. So therefore, this KLB determine the tissue selectivity and the sensitivity to FGF21. So there is this is a summary we made several years ago. So shows this FGF21 exert its pharmacological effect by coordinating the inter-organ communication. So that basically FGF21 can act on the wide adipose tissue and the brown adipose tissue to promote the thermogenesis and the browning and the lipid metabolism. And the most important, I will show later on, it can promote this adiponectin production. And FGF21 can also act on the liver, but the effect is not the direct effect. And also, there are also studies suggesting FGF21 can act in the brain and to possibly to regulate the food intake and then preference for sugar and the alcohol consumption. So, what is the main target tissue of FGF21? And then is the brain or adipose tissue or liver. So our laboratory and others have shown this liver, although it's a major site for FGF21 production, but not the FGF21 actions, because the main receptor, this is FGFR1, is not expressed in the liver tissue. So we find that the adipose tissue is the main target tissue of FGF21. And there, in fact, so in FGF20 at this adipocyte, and we have shown a series of evidence suggesting so this FGFR1 and the beta carousal, that is co-receptor, is highly expressed in both white and the brown adipocyte, and initiate multiple signaling pathways to regulate the glucose uptake and to modulate lipolysis and also to modulate the PPAR gamma activity and also heat dissipation. So there are also studies from other groups show, and so these adipocytes play an obligate role in mediating the metabolic effect of FGF21. So then, for example, if the adipocyte specific depletion of FGFR1 and beta carousal, and then in these mice, the metabolic benefit of FGF21 is abrogated. And similarly, so in this lipodystrophic mice without adipose tissue, this is also resistant to metabolic action of FGF21. So therefore, so we have been asking, how does this FGF21 exert its profound effect on glucose and lipid metabolism and protect against this fatty liver disease? So we have find a serious evidence suggesting to this adiponectin so act as a downstream mediator.
Abbiamo detto dal martedì, ma non domani, quindi da mercoledì poi.
So
No, ma domani comunque non ce la facciamo.
Cristina, you have the mic on. Please shut it down.
Domani non possiamo fare perché non abbiamo raccolto.
FGF21. Okay, so this adiponectin, as you know, is a major adipokines secreted from adipose tissue with insulin sensitizing anti-inflammatory properties. And this, in the circulation, it forms several oligomeric complexes, including this trimer, hexamer, and the high-molecular adiponectin. So this adiponectin acts through its receptor called this ADIP-R1, so and the ADIP-R2. They are 7 transmembrane receptor, but they have an opposite topology with traditional G-protein coupled receptors. So actually in 2003, so when I did my postdoctoral study, so I find this adiponectin can possess potent protective effect against non-alcoholic fatty liver disease. And as you can see from here in this, so the fatty liver, and after treatment with this recombinant adiponectin, this lipid accumulation in the liver is completely depleted. It's a very striking effect. So we find actually about 10 years ago, this FGF21 can actually induce both the expression and the secretion of adiponectin in mouse adipocyte. As you can see from this left panel, this is from primary adipocyte. After treatment with the recombinant FGF21 for 24 hours, the secretion of adiponectin into the culture medium is markedly increased. And then in this adiponectin knockout mice, as you can see from here in the middle panel, is the Y-type mice. So after treated with this FGF21, we see the marked elevation of fatty liver compared to this vehicle control group. However, in this adiponectin knockout mice, even if after FGF21 treatment, and there is no improvement in fatty liver, so suggesting this adiponectin is required for FGF21 actions. Similarly, if you look this ALT, AST, and level, so in the Y-type mice, after treatment with FGF21, we see the decrease in ALT, but not in adiponectin knockout mice. So this, after our publication, after 2 years, this Inanini company also tested their FGF21 analogs in humans in type 2 diabetic patients. They also observed, confirmed our animal study showing after its treatment with the FGF21 in type 2 diabetic patients with obesity, this adiponectin is dose-dependently increased after treatment. And then, in fact, and all the clinical trials on FGF21, and then tested so far, such as these Pfizer compounds and these BMS compounds for these FGF21 analogs, and then also these FGF21 receptor agonists, they all shows this FGF21 treatment can increase adiponectin, and also the increased level is closely associated with the improvement in non-alcoholic fatty liver disease. So therefore, we found a serious evidence suggesting so this FGF21 adiponectin access is they're through mediating the crosstalk between the liver and the adipose tissue, and then contribute to the improvement of metabolic homeostasis and also to the improvement of fatty liver. Basically, FGF21 produced either from the liver tissue or from adipocyte act in an endocrine or paracrine manner on adipose tissue itself and promote adiponectin secretion. And then adiponectin can then travel through the bloodstream to the liver to improve insulin sensitivity, to increase the fatty liver or to decrease fatty liver. Can also act in the muscle to decrease lipid accumulation and improve insulin sensitivity. So, but although FGF21 has multiple therapeutic benefits, but this FGF21, the endogenous FGF21, human FGF21, is not druggable because it has several limitations. One is a very short acting half-life, only 0.5 to 1.5 hours. So when you produce the endogenous FGF21, it's unstable, susceptible to proteinase degradation by this fibroblast activated proteinase called FAP. So this is quickly degraded and the protein becomes inactive. And also there is prone to aggregation and precipitation, and then very hard to scale up. So due to these limitations, And then there are many, so chemical and genetic engineering has been attempted, and then to try to generate non-acting bioactive form of FGF21 agonist or receptor agonist. So this, including these strategies with the FGF21 analogs or FGF21 receptor agonist. So many of these FGF21 analogs has been developed. The initial one developed by Eli Lilly, but now they have stopped because it's not competitive enough. So acting half-life is still very short. And then these BMS compounds, and then it's generated by pagylation, and then linking to this FGF21. These Pfizer compounds, and that is linking FGF21 to FGF21 molecules to this to antibody fragment. And the most promising one is the FGF21 developed by this Aimin company, so currently under the phase 3 clinical trial, and that is linked the antibody FC fragment with 2 FGF21. And there are also other developments such as use the FGF1 and then without the heparin binding domain, and then conjugation with the FGF21 C-terminal to increase its potency. So they're all actually under the various phases of clinical trial. So additionally, there is also FGF21 receptor agonist that has been developed. So there's, for example, this Aimin has developed this called biospecific Aimin. So this is completely as an artificial Aimin that can bind to FGF21 receptor and activate its signaling. And also this Genentech has developed this monoclonal antibodies targeting FGF receptor R1 only. And then there are also many of these hybrid to receptor monoagonistic antibodies that bind to both receptor and its co-receptor KLB1. And among these, for example, this genetic one, BFK, this is called this, so the beta-carozo FGFR1 dual receptor antibody agonist. So this is right now under phase 2 clinical trial. So that we actually has also developed ultra non-acting FGF21 analog by this lipidation technology. So you can do so every injection for every 2 weeks. So we did it by using this translipidation reaction by adding this carbon 16 to fatty acid to this lysine residue, and then this lysine residue will bind to this human albumin, quite similar to what the Novologix did for this semaglutide for the GLP-1 agonist. And then this is but now under the phase 2 clinical trial in both China and the United States. And then as you can see from here, and then we see that after this treatment for this lipidated FGF21, so you see the market improvement is even better than this FC-tagged FGF21. If you use the endogenous FGF21, and we see a very mild effect in terms of fatty liver accumulation. So there are over 20 registered clinical trials for these FGF21-based drugs for treatment of obesity-related metabolic syndromes. And this is just a few examples, just showing these BMS compounds. This is pegylated FGF21 in 2019. They are in phase 2 clinical trial. And this occurring one, and that was now they are moved to phase 3 and then clinical trial for that. And this just recent, we did a summary about the recent 3 years and these clinical trials. They show all this has consistently shown the potent therapeutic benefit on this MAFLD and the related complications. For example, this phase 2 clinical trial on this, so the pegylated FGF21 shows improvement in liver fibrosis and the liver injury and the liver inflammation in biopsy proven the patients. And this, again, like this phase 2b trials also shows improvement in NAFLD score and the liver stiffness and the steatosis. And then there actually many they are in the phase 3, phase 2 clinical trials. So quite similar observations on improvement of NAFLD and the improvement in lipid profile. So that is FGF21. So more recently, so we have find another liver secreted hepatokines called autotoxin. So this autotoxin, we find this also play a role in regulating this, contribute to this MAFLD formation, actually by suppressing this FGF21. So basically during the obesity, so this autotoxin is increased and then suppress the liver to produce FGF21 and then increase the fatty liver. So what do we find actually is from, so this is the autotoxin. So autotoxin is a circulating enzyme that catalyzes the generation of the LPA, lysophosphatidylic acid A, so LPA from the LPC. So once you convert this LPC is converted to LPA, this can activate the LPA receptor. So LPA receptor, they are gibberellin coupled receptor. So once that is activated, so this will activate multiple inflammatory pathways, trigger this inflammation and also fibrosis. So in our clinical study, so we have found this serine autotoxin is actually biomarker closely associated with the liver dysfunction and the MAFLD severity in obese Chinese. As you can see from here, this hepatic fat content is closely associated with the serine autotoxin level, also with the ALT/AST level. And also this autotoxin level is associated with the histological severity of this NAFLD in biopsy-proven patients, including this inflammation, ballooning, fibrosis, and also histological diagnosis. So that in animal models, so we find quite similar the trend. As you can see from here, after high-fat diet, the serine autotoxin level, this yellow one, so that is increased progressively. And then also the autotoxin enzymatic activity also increased progressively. So we find by this profiling, tissue profiling, find this increase mainly is in the liver tissue. And when we do the further cellular fractionation analysis, we find the high-fat diet induced increase in autotoxin occurs predominantly in hepatocyte, but not in other tissue. So that if we use the knockdown the hepatic autotoxin, and you can see from here this high-fat diet induce the fatty liver, and then if we knock down the hepatic expression of this the autotoxin using the adenovirus associated virus approach, and this the fatty liver is decreased. And then after that, so we have developed two highly specific high affinity anti-autotoxin antibody. So basically by targeting this hydrophobic binding pocket for this autotoxin. Remember, this autotoxin is an enzyme, so that bind to LPC. So we make the monoclonal antibody targeting to this binding pocket, and then this can suppress the enzymatic activity of this autotoxin. And then using this method, autotoxin neutralizing antibody, as you can see from here, and after the treatment, this is a control after high-fat diet in the right panel. So after oil-ready oat staining, you can see the huge lipid accumulation, and this autotoxin antibody neutralizing antibody can decrease the fatty liver accumulation, and then also confirmed by this biochemical analysis of liver triglyceride, and this ALT, AST, they all decreased. And then most importantly, so if we neutralize this autotoxin, you can see the fatty acid oxidation, that is FAO, that is also increased in the this treatment group. And then mechanistically, so we find this FGF20, if we neutralize it with autotoxin, actually this can increase FGF21 selectively in the liver tissue and the adiponectin. So we have measured all these different hepatokines and adipokines, including this angiopoietin like protein 3, 4, 6, and the futin A, FGF1 like DPP4, lipokinin 2, naptin, all this, there's no change. But we see there is a marked increase in FGF21, serum FGF21 level after autotoxin antibody treatment. And the mRNA expression in the liver tissue is also increased dramatically, and also adiponectin that is decreased, also increased. So, and then on the other hand, if in this FGF21 deficient mice, if we treat with this anti-autotoxin antibody, and the therapeutic benefit of anti-autotoxin antibody is reduced or disappeared, as you can see from here in the wild-type mice. And then after treatment with autotoxin antibody, and the fatty liver is reduced, but the FGF21 knockout mice, and then there's no effect. Similarity, and this ALT level, AST level in wild-type mice, we see the obvious decrease after FGF21 treatment or after anti-autotoxin antibody treatment, but not in this FGF21 knockout mice. So therefore, and we also find this autotoxin can suppress hepatic FGF21 production by modulating this LPA, that is the substrate, and the ERK and PPAR alpha signaling pathway. I'm not going to go this in detail. So that is in summary. So we find in obesity status, so this hepatocyte production of this autotoxin is 80x, and the secretion is increased. And then this acts in an autocrine manner in the hepatocyte to so produce this LPA, and then act through this LPA receptor, that is G-protein coupled receptor, and then activate this ERK signaling pathway. And the ERK then, through the phosphorylation dependent inactivation of PPAR alpha, and then suppress PPAR alpha activation. And then because FGF21 can be activated by PPAR alpha, so once the PPAR alpha is inactivated, the production of FGF21 is reduced, and then the effect of FGF21 acts on adipose tissue to reduce to produce adiponectin. That effect is also reduced, and then contribute to steatosis, inflammation, and fibrosis. So on the other hand, if we use this blue with this anti-autotoxin antibody, neutralizing antibody, so we can reverse the entire process. So therefore, so biopharmaceutical intervention to suppress autotoxin and or to increase FGF21 to represent a highly promising remedy for this obesity-related MAFLD and also liver fibrosis. In the last couple of minutes, I just want to talk our recent finding to find a new biomarkers for MASH. So we have established using this ONINC system. So that is based on Switzerland. So you can use this proximity extension assay to measure over 5,000 proteins in 5 microliter of serum samples. So using this technology, so we have identified this serum secreted CPCT1 as a top-ranked biomarker for MASH. And then as you can see from here, and then there is this CPCT1. This is clearly the top candidate. This can clearly differentiate the patients from NASH with the simple steatosis and also normal liver. So this is in this situation, as you can see from here, and in NASH, there are several fold increase and then in secreted CPCT1. And this CPCT1 and the full name is the carb domain containing protein 1. And this is actually the single transmembrane protein, but the secreted CPCT1 here that can be cleaved by the proteases such as plasmin and also TPA or other proteases, and then released into the circulation. And then we have developed this highly robust assay for quantification of secreted CPCT1. And then we find, as you can see from here, we did the validation in both our main cohort that is based on the hospital in Guangzhou that with 489 biopsy-proven samples with normal liver, non-alcoholic fatty liver, and the NASH. And then we have also collected about 135 external validation cohort. As you can see in both these cohort, and this CPCT1 can clearly distinguish this NASH from this NAFL and also normal liver. And also this is closely associated with fibrosis, and then these patients with advanced fibrosis, and also significant fibrosis from F2 and F3 in both this main cohort and the validation cohort, and that can clearly higher than this without fibrosis. And then in this bariatric surgery patients, and then we have done this up to 24 months follow-up study. And then as you can see from here, and in the bariatric, after bariatric surgery, and then this in those patients with NASH, and then after surgery, and this secreted CPCT1 is obviously decreased. And then in those with non-MASH, and there's no obvious changes. So this suggesting this marker not only can be used for the diagnosis potential risk screening of MASH or NASH, but also can be used for therapeutic monitoring of this MASH. So we also established this called C-dash score using machine learning. As you can see from here, we have collected 104 those different clinical biochemical parameters, and together with the serum CPCT1. And then after using the AI, we have established this C-dash score, DAG score, that including the secreted CPCT1, diabetes, AST, and the gender. And then these, using this score, so we can clearly find, so you can see using the C-DAG score for MASH, we can get the AUC of around 0.9 in this, our discovery cohort, and also in about 0.89 and in this validation cohort. It's a very robust biomarker, and we are trying to validated independently by collaboration with others. And then this CPCT1 is also pathologically involved in the NASH development. So we have done two different animal models. One is choline deficient high-fat, high-cholesterol diet. Another is high-fat, high-cholesterol diet, as you can see from here. So then these the wild-type mice, after this diet, they develop the NASH, develop fibrosis, and then in these knockout mice, the development is reduced. This is quite similar model in high-fat, high-cholesterol diet. So, so this is a circulating CPCT1. Certainly is a top-ranked classifier of MASH by owning proteomics analysis. The decrease in CPCT1 mirrors changes in liver enzymes after bariatric surgery. So we have established this CPCT1-based algorithm, and it shows this is excellent performance for risk stratification of NASH. So that is pretty much of my talk today. Finally, I want to acknowledge my collaborators in Kyoto University and for FGA21 study, and also several centers in mainland China, such as Shanghai Diabetes Center, and then also Xiangya Hospital for collecting all these clinical samples for Wenzhou Medical University, and then they on FGA21 pharmacological studies. Thank you very much for your attention. This is actually the light view in Hong Kong, in Victoria Harbour. So I hope in the future you can visit here, like we can enjoy the light view here. Thank you.
Thank you very much, Dr. Xu, for your presentation. It was really clear and very easy to follow all your data and your ideas. So congratulations for your presentation.